Hello friends, I welcome you to my YouTube channel Govind Narayan Purohit, wherein we are discussing the various topics of theriogenology, and I am highly thankful to you if you have already subscribed to my channel. If you have not subscribed to my channel, kindly do not forget to press the subscribe button and press the bell icon. Please turn on the notifications so that you get the information of our next video very fast. Friends, you can scroll through the playlist of our channel and you can just see the videos of your interest, right? So in our today's video we discuss about the practical procedures in vitro fertilization, right? Now, what are the basics? How in vitro fertilization is being done? You see, for in vitro fertilization, the very first thing you have to recover the oocyte, and then because the follicular oocytes in most domestic mammalian species are immature, so they are put to in vitro maturation. After maturation, the uh, oocytes are fertilized with prepared sperms and then they are put to culture for a period of 7 days when they develop into blastocyst. Uh, then they can be used for the purpose that one intends to. Now, what are the basic steps for in vitro fertilization? The basic steps involve the oocyte collection, the in vitro maturation of the eggs, the in vitro fertilization of the eggs, and then in vitro culture of the embryos or the presumptive zygotes, and then the embryo transfer, right? Now, for oocyte collection, there are various methods, but the surgical removal from a cow, but that's not really a practical procedure. So, they can be removed from oocytes after slaughter, and most oocytes from most domestic mammalian species remain viable for a period of 12 hours after death. So, that can be another approach, or you can retrieve the slaughterhouse ovaries, and then from the slaughterhouse ovaries, as you can see in these pictures, the oocytes are retrieved. Then another way of retrieving the oocytes from live animals is transvaginal ultrasound guided ovum pickup. For this purpose, there is a ultrasound which has a long needle attached to it, and then the oocytes can be retrieved from the live animals, visualizing the follicle and then puncturing the follicle and retrieving the oocyte. Oocyte in vitro fertilization has become popular because a large number of embryos are required for many empirical studies, and so for those studies, usually many laboratories they develop embryos by using the abatoir derived ovaries, right? Now then, from a live animal, oocytes can be recovered. There is a ultrasound probe, which is a sector probe, and it has got a long needle. You can see the needle over here, and it's very long. And the ultrasound probe is introduced in the vagina. Just ahead of the vagina is the peritoneum, and one hand, a person keeps his hand in the rectum and catches hold of the ovary. You can visualize the follicle on the ovary and just pierce the peritoneum to so that the needle reaches near the ovary, and then when you just pierce it, then you can just aspirate the contents by negative pressure, and the follicular fluid will come outside along with the oocyte. So this can be performed in dairy cows using a 18 gauge needle and 100 to 200 mm of pressure, and then OPU can be performed once to twice weekly depending on the species and depending on the efficiency of the operator, right? Then oocyte aspiration uh, is the method of choice for retrieving oocytes from slaughterhouse ovaries. And I will just show you how the ovaries are aspirated in this video clip. You can see the every work should be done and in a laminar airflow, and the ovaries have been collected in the medium, and the ovaries caught hold, and the needle is taken, and then just it is punctured, and the follicular fluid is aspirated by negative pressure, and you can just collect the oocyte. Now you can see a lot of oocytes on the ovaries, and you can collect each one of them by rotating the needle while being inside the ovary, and be careful not to pierce the ovary, and you can see that when the oocyte is, the follicle is aspirated, the follicular fluid, it comes inside, and there is a depression on the ovary, and this in this way you can just collect the oocytes by follicular aspiration of the oocytes in the laboratory. But for collecting the oocytes, you have to be careful, and then there are other methods of ovarian slicing, and they can also be used for collecting the oocytes. Then when you just aspirate the follicle, there is a depression on the follicle, as you can see in this picture, and this shows that yes, now the follicle has been aspirated. After aspiration, the oocytes are kept in drops, and then one can give serial washings, and these washings can be given by moving the oocytes, visualizing under a microscope, and moving the oocytes from one drop to another, then to next, then to next, and then to next. So good quality of oocytes, 8 to 10, are placed in culture drops of 50 to 100 microliter in vitro maturation medium in small petri dishes, 35 or maybe if you are using some different type of material, you can also use those multi-well plates, or you can use smaller dishes or larger dishes. And the oocyte, the identification of the oocyte is that it has got a clear cut zona pellucida, and many of them have the cumulus cells attached to it. Then we have to select good quality oocytes. The good quality oocytes are those which have 3 to 4 or more layers of the cumulus cells attached to it, and you can see in these pictures that these oocytes have 3 to 4 or more layers of cumulus cells attached to them, evenly granulated cytoplasm without breakage of the zona pellucida. These are some of the peculiarities for the good quality oocytes, right? Then poor quality oocytes, you can find most of them do not have a zona pellucida, they are denuded, or maybe they are distorted in shape, or maybe they are having uneven granulations in the cytoplasm or breakage of the zona pellucida. So these types of oocytes are not to be used for in vitro maturation and further in vitro fertilization, right? Then oocyte searching and washing is done under a microscope. Once you have just collected, you just search the oocytes under a microscope. You can use a micropipette, you can use a pasture pipette, and then you give serial washings to the oocytes. They are taken from one drop to another and then to another, so that the tissue debris attached to the oocyte is removed, and oocytes are free of any tissue debris. Then you can, after selecting the oocytes of good quality, then you can place them in one culture drop. You can see here it is being placed in another dish, and in this dish you can place the oocytes, and then you can prepare the micro drops, and these micro drops, they are maybe from 50 to 100 microliters, and the micro drops, they are placed on the dish, and then over the micro drop of 50 to 100 microliter we place paraffin oil, so that when the oocytes are placed in the CO2 incubator, they do not dry up. So it has to be taken in mind, and then the oocytes, they have to be kept in groups of 5 to 10 or maybe maximum 15 in one culture drop, of 50 to 100 microliter, and then they are covered with paraffin oil. A lid is capped over it. They are labeled, and you mention the all labels of that oocyte, and then you can place them in the CO2 incubator. The oocytes are placed in the CO2 incubator for in vitro maturation. The time frame for different species varies, and you have to place them in the CO2 incubator. A CO2 incubator has a temperature of 38.5 plus minus 1 degree centigrade, CO2 tension because of the CO2 cylinder attached to it, and then underneath there is placement of the sterile distilled water, which maintains a relative humidity of 80 or more percent, whatever you want to keep for your culture program. Then after the oocytes have been kept for 24 hours, and this time frame varies from species to species and from lab to lab. The oocytes are taken out, and then they are evaluated for the maturation. Now, for studies, you need to evaluate the maturation status. Otherwise, for IVF lab, you can take out 10% of the oocytes, the total oocytes that you have placed for maturation, and then evaluate them for the maturation. Oocytes can be evaluated for maturation by two types: the cytoplasmic maturation, which you can see here, the cumulus cells have expanded, and then for nuclear maturation, you can stain them with the Orsin stain, and then examine for the nuclear status of the oocytes, right? So here you can see that the oocytes that have attained the cytoplasmic maturation, the cumulus cells will separate away from uh, the zona pellucida, and they will move further. And then you can see in these oocytes the cumulus cells have not moved away. Here in this picture you can see the cumulus cells have moved away. Again, this one, cumulus cells have moved away. Then another could be, if you find a oocyte with one polar body extruded, then that shows that the oocyte is mature. And then for staining of the oocytes, only 10% of the oocytes are taken out for staining. The oocytes are taken out, and then they are vortex agitated, and by repeated pipetting, or by vortex agitation, or by placing a drop of KCl long in a tube in which the oocytes are placed, and then repeated pipetting will separate out the cumulus cells, and the oocytes will become denuded. Now the denuded oocytes are fixed on by placing them on a slide and fixed in cold acetic methanol and stained with acetoorsin. Now you can see on this drop the oocytes are there, and then for fixing we will take the oocytes with a minimum possible volume and place them on a on a slide with a minimum of volume, so that when you fix the oocytes, that volume does not propel the oocytes towards the sides of the slide, and the oocytes remain in the center where you intend them to be there. So then oocytes are placed a minimum drop of fluid, and then a cover slip is applied, and then these oocytes are this slides are fixed on all the sides, and then these slides are dipped in the acetic methanol and kept for 24 hours so that they become fixed. After fixation, the slides are taken out, and then they are stained with acetoorsin, or maybe you can even use GNP. Then when they are examined after staining, when they are examined under the microscope, you find very beautiful pictures of the oocytes. You can see here, this is a GV stage oocyte. There is a nucleus, nucleolus, and the germinal vesicle is still there. Then in this one you can see this is stained with GNP, this one is stained with orsin. You can find the condensation of the chromatin material and the formation of the tetrad that marks the GVBD. And then in this one you can find that the anatelophase, the chromosomes have started separating further, and this gives very beautiful pictures wherein you can evaluate the nuclear maturation of the oocytes, right? Then the better stage after maturation for 24 hours is the metaphase 2 plate. This is the metaphase 2 plate, and then this is the polar body released, and this is the metaphase 2 plate or the spindle. Now this is the metaphase 1 oocytes, and after evaluation, then you can note down the percentages of the oocytes that have reached the different stages of maturation, right? Then in a in vitro fertilization laboratory, the important aspect is the preparation of the sperm, and the spermatozoa has to be prepared, because in natural mating the sperm traverses the female tract and becomes capacitated and attains many changes. Now the seminal plasma is left behind, so this you have to do artificially for preparing the sperm for in vitro fertilization. Now, for the purpose, you have to remove the seminal plasma, and you have to select the motile sperms, because the seminal plasma hinders the motility of the sperm. So, there are two approaches. The most common approach for the most domestic animal species is the Percol gradient separation, although it is not good for a few species, but usually Percol gradient separation is being used. What is being done? The 90% Percol and 45% Percol, or maybe some labs are using 80% and 40% Percol, and you can see here a semen straw is emptied over here, and then it is centrifuged, and then the motile sperm, they traverse, they pierce the Percol layer and reach to the bottom. And so the most motile and most active spermatozoa will be here, and the seminal plasma will be left above in the supernatant. That is discarded. This is again redissolved, and this there will be formation of a sperm pellet. You can see here in this test tube a sperm pellet has been formed here. This sperm pellet has the most motile sperms, and so then you can select these sperms for capacitation. Now in Percol gradient, this is again showing the Percol gradient centrifugation, and after centrifugation the motile sperms are in the lower level of the test tube. So when you are taking out spermatozoa, you will have to pick them up from the lower level, right? Then for swim up procedures, which are used for some species and for human in vitro fertilization, the sperm is added with a medium, and then it is centrifuged, and then it is allowed to stand. The seminal plasma is removed. The most motile sperms will swim up in the medium, and then you can select the motile sperms from the upper layer of the medium, because the motile sperms move up in the medium, right? Then sperm need to be capacitated. There are a variety of capacitating agents. The most common one is heparin, and for increasing the motility of the sperm, phenylephrine, hypotaurine, epinephrine mixture, or caffeine mixture, or talc, they are used, biomedium. And then for capacitation, the spermatozoa concentration is checked, and then they are kept in a CO2 incubator for 2 to 6 hours before insemination, so they attain capacitation, and then one can use this spermatozoa for insemination. Now when the oocytes have matured, you have kept them for 24 hours, you take out the when you have already prepared the spermatozoa, it's ready. You can check the spermatozoa whether they are capacitated spermatozoa become hyperactive and they move very fast. You can check them under a microscope, and then when you have already done all this, and you can take out the sperm suspension. And for the oocytes which are kept in 100 microliter droplets, you take out around 80 to 80 microliter of the medium and introduce the sperm suspension 80 microliter. Now you have inseminated, or rather you have introduced the spermatozoa inside the medium in which the oocytes are kept. Again, you cover it with paraffin oil and then apply a lid, note down or mark what has been done on what date using what sperm, and then again place them in a CO2 incubator. Then allow the sperm and oocyte to co-incubate for 6 to 18 hours in a CO2 incubator. You can see here the sperm and oocytes are co-incubated, and you can see under a microscope that the spermatozoa are moving very fast and they are ready to fertilize the oocyte. After fertilization, if one is going in for some study, he can evaluate the status of fertilization by taking out again 10% of the presumptive zygotes and fixing them in a way similar how the oocytes were fixed for evaluating nuclear maturation. And then one can find either a metaphase 2 plate and a sperm head, or one can find male and the female pronuclei, or the pronuclei intermingled and the zygote is complete, and one can also visualize the two polar bodies that have been extruded. So that way one can evaluate fertilization. Otherwise, if you don't want to evaluate the fertilization, then what can be done? Sperm suspension is removed and the presumptive zygotes are washed and moved to the in vitro culture medium. The in vitro culture medium varies from species to species, and you just take out the sperm suspension, add the culture medium, and again take out the medium again add fresh medium, and then again add paraffin oil over it and cover it with a lid and place them in the CO2 incubator. Now, when you are placing the oocytes for culture for a period of 7 days, then usually we have to add a cell line, because there is a culture induced developmental block at the 2 to 4 cell division of the oocytes. So to avoid that, some cell line, maybe granulosa cells or cumulus cells, or maybe any other cell line, buffalo oviductal epithelial cells or the bovine oviductal epithelial cells, they can be collected very easily. We just cut the two ends of the oviduct, clamp it on one side, and then squeeze it using a forceps to take out the cells in a culture dish. And then you can add medium, and because there will be a lot many cells, so you have to take a small amount and then place it in fresh medium, add fresh medium, and then do repeated pipetting to separate out those cells, and this cell line will then be added to the presumptive zygote that are being cultured. And after 24 to 48 hours or maybe later, it will form a monolayer, and this will help the oocytes to or the presumptive zygotes to reach the blastocyst stage, right? Then the dishes are again incubated for 7 days in an incubator and are checked periodically for embryo cleavage, the cell division, and you must replace the medium every day, because cells in culture will consume whatever is available in that medium. So to assure the nutrition of the presumptive zygotes, you have to change the medium every day. And after 7 days, for 7 to 8 days, if you find these type, this is a unfertilized ova, it has not grown, this is a ova that has cleaved 2 cell or 4 cell, or but the cells are uneven. Some cells are large, some of them are small. So these are not the good quality zygotes that have resulted. Again, this one, after 7 days, if you find 2 cell egg, then again this is not a very good option. This is a morula stage embryo, but you can continue to keep such type of embryos in culture for a further period, so that to see whether it develops into a blastocyst or not. Then after 2 to 3 days, when you evaluate, and if you find the embryos with two cells, then it's all right. But after the end of the culture, if you find embryos like this, they are not very good, right? The end result should be blastocyst. Blastocyst, you can see expanded blastocysts are there. Some of the blastocysts, because a blastocele appears inside, and these blastocysts would be good for transfer. They will be good for use in studies, and you can find a group of embryos that have developed, some of them blastocysts, some of the later stages, or some maybe are still in the hatch. A few might be a hatching blastocyst, as you can see this one, right? So this is what is desired as the end result in vitro fertilization. Now this table, it shows the peculiarities of, or the media, or the time frame, or the different sperm concentration used in vitro fertilization of different species. For the bovine species, for IVM media, TCM 199, medium SOF, Hemzaptan, all can be used. Maturation time is 18 to 24 hours. The supplements are usually FSH, LH, estradiol, and bovine serum albumin, or maybe estrus cow serum. And then the medium for IVF is either talc or biomedium, and sperm preparation is by swim up or percol, and then sperm motility enhancement is PHE or caffeine, and capacitation is by using heparin. We require the 2 to 10 million sperms for insemination, and sperm oocyte co-incubation time varies from 2 to 10 hours. Similarly, for the different species, especially for the pigs, the mediums used are different. NCSU2, 3, and BECM6, medium weymouth, and they are kept for maturation for 38 hours. And then the medium used for IUF is MTBM or talc, and then caffeine is used, and sperm capacitation is by cysteine, and it requires 2 million spermatozoa, and the sperm oocyte co-incubation is hardly 4 to 8 hours. So likewise, there are different suggestions based on many studies carried out in different species, and one can use these when working with a particular species, when one can use the most appropriate medium and most appropriate approaches, right? Now, the success rate of the IVF varies widely between protocols, culture conditions, and the different labs. IVF success rates are the percentage of all IVF procedures which result in a favorable outcome, which implies the pregnancy rate or the live birth rate. But in some labs, they consider the blastocyst rate as the success rate. Due to advancement in reproductive technology, the IVF success rates are substantially better and today than they were a few years ago. So, and when you are working continuously, you continuously improve your working efficiency and the blastocyst rate also. Now, what are the limitations? The limitations of IVF are you can introduce some genetic defect in oocytes, and you can there can be genetic defects in fertilizing sperm. There can be environmental mutagenesis, inadequate supply of nutrients and hormones, and exposure to toxic agents and free radicals. These are some of the limitations of the IVF. Then another limitation of IVF is when the oocyte is kept in a medium, then the cells grow faster, and so there is a large calf syndrome. The calf grows at a rate faster than the normal calf, so that is one of the problem of in vitro fertilization, right? So these are my contact details: gnpobs@gmail.com. You can contact me anytime. This is my YouTube link, and if you like the video, kindly give your likes, kindly share the video, kindly give your comments, kindly do not forget to subscribe to my channel and turn on the notification button. And if you have any query, any suggestion, you are welcome to give it at my email gnpobs@gmail.com. So have a nice day. Have very good results in your IVF experiments. Thank you. Bye bye.